What do epigenetic changes reveal about CFS-ME biomarkers? Imagine your genes are like a vast library of instructions, but how those instructions are used can change without rewriting the books. That's what epigenetics is all about. In chronic fatigue syndrome and myalgic encephalomyelitis, or CFS and ME, scientists have found that these epigenetic changes can tell us a lot about what's happening inside the body. Specifically, they look at patterns of DNA methylation, which is a chemical modification that can turn genes on or off without changing the actual genetic code. These patterns are different in people with CFS or ME compared to healthy individuals. Research shows that these DNA methylation differences are widespread, especially in immune cells. Advanced techniques have identified tens of thousands of sites across the genome where methylation levels differ. Many of these sites involve hypomethylation, meaning there's less methylation than usual. When methylation decreases, it can lead to increased activity of certain genes. These genes are often involved in immune responses, energy production, brain function, and metabolism. This suggests that the epigenetic changes may contribute to the main symptoms of CFS and ME, like immune system problems, fatigue, and cognitive issues. Interestingly, these methylation patterns are not static. They can fluctuate during disease flare-ups, which means they are sensitive to changes in disease activity. This makes them promising as biomarkers or biological indicators that could help doctors monitor how the disease progresses or how well a treatment is working. Some specific genes that are often affected include brain-derived neurotrophic factor, which influences nerve health and pain, and catecholomethyltransferase, which plays a role in managing inflammation. Researchers have also found that genetic variations, called single nucleotide polymorphisms, in immune receptor genes often occur alongside these epigenetic changes, showing a complex interaction between genetics and gene regulation in CFS and ME. From a practical standpoint, these epigenetic biomarkers could make diagnosis more precise by providing objective molecular signatures that distinguish CFS or ME from other conditions. They might also help tailor treatments to individual patients by identifying specific molecular targets and tracking disease activity over time. However, it's important to remember that these findings are still being validated. Larger studies are needed to confirm how reliable these epigenetic patterns are across diverse patient groups and how they influence symptoms and disease course. In essence, the study of epigenetic changes in CFS and ME reveals that the illness involves complex shifts in gene regulation affecting immune, metabolic, and neurological systems. These changes offer exciting possibilities for improving diagnosis and understanding the biological roots of these conditions. Still, ongoing research is crucial to turn these insights into practical tools that can truly help patients. If you or someone you know is dealing with symptoms of CFS or ME, it's always best to consult healthcare professionals who can provide personalized advice based on the latest scientific knowledge.